Welcome to a mini-series of Neurosalience, featuring interviews with some of this year's annual meeting's keynote speakers.
Hello, welcome to the OHBM 2023 keynote speaker interview. I'm Xinhui from the OHBM Communication Committee. Today, it is our great pleasure to have Dr. Hongkui Zeng here to share her experiences and insights. Hongkui is Executive Vice President and Director of Allen Institute for Brain Science. Since she joined the Allen Institute, she has led several efforts to develop and operate high-throughput pipelines to generate large-scale and open-access datasets and tools to accelerate neuroscience discovery. Her current research interests are in understanding neuronal diversity and connectivity in the mouse brain-wide circuits and how different cell types work together to process and transform information. Through her leadership of multiple scientific teams at the Allen Institute, she has built several research programs using transcriptomic, connectomic, and multimodal approaches to characterize and classify the wide variety of cell types that constitute the mammalian brain. Her work has led to widely adopted community resources and standards, including transgenic mouse lines, Allen Mouse Brain Connectivity Atlas, the Common Coordinate Framework, and the brain-wide transcriptomic cell type taxonomy. Hongkui received her PhD in molecular and cell biology from Brandeis University, and she did her postdoc at MIT. She has received many honors and served on multiple committees and advisory boards. Welcome, Hongkui. It's great to have you here.
Thank you very much. Thanks for that nice introduction.
I'm curious about your academic trajectory. From your bio, your PhD focused on studying the molecular mechanism of the circadian clock in fruit flies. Then you investigated the molecular and synaptic mechanisms underlying hippocampus-dependent plasticity and learning as a postdoc. Could you tell us a little bit what motivated you to work on these projects? What triggered your interest in neuroscience?
Yeah, my original background from my undergraduate work is, very much on biochemistry. My degree was in biochemistry, so my background is more on the molecular and biochemical aspects of biology. When I went to Brandeis University for my PhD, first of all, there's a very strong neuroscience program. Just talking to the faculty members, I became a lot more interested in how brain works. And I started with the Drosophila circadian rhythm research, because it's really a great combination of my molecular background with genetics and then getting into a little bit about the brain itself. So I feel that I would like to move beyond just, studying the brain from the molecular genetic point, but also to link it with the physiology and behavior. So that's why for my postdoc, then I switched from Drosophila to mouse as a model system and continue to use genetic approaches, but then to look at the roles of genes in driving plasticity, neuroplasticity, and also in regulating behavior.
After your postdoc, you joined the Allen Institute in 2006. Could you please share what your research career trajectory has been like at the Allen Institute?
I have been at the Allen Institute for more than 16 years now. I started with my own little group working with me on the genetic tools project, which was, how I started at the Allen Institute using my genetic background and cell type specific manipulation, kind of skills. So from there, my team grew from 2 people to 10 and eventually to a 25 people group. And the projects that we did also expanded from creating transgenic mouse lines to using AAV viral vectors to map connections, projections, and connections in the brain, and then to beginning to explore new technologies to characterize cell types and single cells in different parts of the brain. That also went along with the expansion of research programs at the Allen Institute. And at the Institute, we expanded our technology platforms to incorporate single-cell transcriptomics, neural anatomy, both mapping connections at both population level and a single-cell level, and then continuing to add new pipelines and new capabilities. My team has been working on more projects as well. I think I displayed this capability of being able to run large-scale projects very well. So then in 2016, 10 years after I worked at the Institute, I was promoted to be the executive director of the structured science division. So, moving from running a lab or my individual research team of about 25 people at a time into running an entire division, which has about 130 people. So so my responsibility increased dramatically at that time. And then in 2020, I was promoted again into the executive vice president and director of the entire institute, Allen Institute for Brain Science. So that was another jump for me in terms of playing leadership roles again, from managing 130 and 140 people to managing an institute of 250 people. I felt really fortunate to have this kind of extraordinary opportunity to do big science and team science, and to address important biological questions in a systematic manner.
Now you are leading multiple projects at the Allen Institute, including Transgenic Technology Program, the Human Cortex Gene Survey Project, the Allen Mouse Brain Connectivity Atlas Project, and the Mouse Cell Types and Connectivity Program. Do you find it challenging to coordinate so many large-scale projects? And how do you manage to solve these challenges?
It is very challenging, especially from the beginning, but I think the fortunate thing for me is that I didn't just jump into large-scale projects right away. Everything was built, from scratch. It went from small to big, and I really learned from the process. It's like the Chinese saying, right? Thousands of miles of journey starting with the first step, right?
Right.
It's really like that, one step at a time. I think the critical thing that I realized, and maybe this is something that I'm very good at, which is 3 points. One is that being able to see the big picture. Why do we do this? What are the most critical components? What it takes to get this done? You have to have a clear big picture in mind in order to begin to organize and manage this kind of large projects. And the second is that you need to be able to prioritize. Step one needs to be done in order to enable step two, but also the mutual dependencies of the different components. So able to prioritize and plan so that things can move forward efficiently. That's number two. And then number three is it's very important, being able to simplify the process. It's large-scale project involving many different teams, many different components, but you need to identify the key point and simplify the process as much as possible and convey that simplified version to people so that people don't get confused by the complexity. It is definitely a learning process. I really learned from the experience of doing all these different projects.
Could you tell us a little bit more about the exciting projects that you are currently working on at the Allen Institute?
The central question that we are focusing now is really on understanding the diversity of the different types of cells that comprise the brain. This is our central focus now. We have a series of questions we would like to address. For example, what is the cell type? How many types of cells there are in the brain? And then where do the cell types come from? How are they formed during development? What are the key factors during the development that define the cell type identity, right? So that's related to then the developmental origins of cell types. And then we also are interested in understanding if cell types are conserved between species, especially, for example, between mouse and human, right? Does the human brain have the same set of cell types compared to mouse? Does it have unique cell types of its own? Where are the similarities and where are the differences? The uniqueness of the human brain itself must come from the cell types or their interactions, the networks, right? So we wanted to do this kind of a cross-species comparison. And then finally, the important question is that it's not sufficient to just have a list of cell types. We want you to know what they do, what the cell types do, and how they work together to generate the function, how they contribute to the different kinds of behaviors that brain control.
Yeah, I also noticed that last month your team at the Institute published a preprint entitled "A High-Resolution Transcriptomic and Spatial Atlas of Cell Types in the Whole Mouse Brain." It is very exciting to see this work out. Could you please comment on the potential impact of this work? How will this mouse cell type atlas help us to develop a better understanding of mammalian brains?
Yeah, we are very excited about this project. It is indeed. I don't want to appear to be bragging about it, but I truly believe, and we, our team truly believe that this is a milestone landmark study in creating a high resolution cell type atlas for the entire brain. For the first time in a mammalian species, in a mammalian brain. It's a very comprehensive catalog of cell types. It provides a reference atlas for the entire neuroscience community so that people can relate their work on specific circuits and cell types within that circuit into our reference atlas. They can use our reference atlas to look at genes expressed in specific cell types. From there, infer the potential functions of the cell types. People can also use our data reference atlas to create genetic tools to target specific cell types to study their function. People can use our reference atlas as a baseline to look for gene expression changes that may be happening under different behavior conditions and especially under different diseased conditions. As I mentioned earlier, people can also use this as a reference atlas to begin to compare with other species, for example, with the human brain or non-human primate brains. There are a lot of utilities, there are a lot of implications, and we are building a technology platform, online platform, to present this entire data set to allow the research community to mine our data in a much easier way. So so all of this is really to facilitate the research across the entire neuroscience field. And we are very, very happy that we got this done. It's a tremendous amount of work.
Yeah, you mentioned the how you can relate it eventually to the human brain, right?
Yeah.
What are the challenges there, right? So obviously a lot of this work has been done in mouse. There are a ton of tools available for investigating mouse brain. Eventually, we would like to see some of this done in the human brain. So what are the big challenges you see there?
Well, the first challenge, of course, is the scale. We need to further scale up the work in the human brain. The human brain is 2,000 times larger than the mouse brain. So there are a lot more cells. However, we don't know if there are a lot more cell types or not. From the existing studies comparing individual brain regions between mouse and human, we actually find that the major cell types in a particular brain region are quite similar to each other. So, but still, that's just what the part that we know, right? There could be parts of the human brain that we don't know yet. So we have to scale up the profiling, the omics approaches to be able to have sufficient sampling of the entire human brain. So that's the first challenge. The second challenge is to perform meaningful cross-species comparisons to really identify homologous sets of cell types for cross-species comparison, it's really important to compare at all these different hierarchical levels. From what we have known so far, we believe that at the major branch level, the cell classes are probably very well conserved and they are easily identifiable. And then from there, you go deeper and deeper and see how far you can go. Then the third challenge would be the variations, technical variations, and especially individual variations in the human population. That's a major challenge coming. In the mouse, we study an inbred strain, genetically identical inbred strain. There's not so much cross-animal variations, but when we go to the human population, there's tremendous genetic variations, and then also, variations at structural and functional levels. So how to reconcile that, it's going to be a major challenge as well. But that's also the interesting and the important part, right, about the human brain.
Yeah, thank you. You've hinted at a bunch of extremely rich questions and studies over the next number of years, and sort of laid out how you've developed your thinking and about how you can actually tackle some of these. What's sort of the time scale you're working at, or in the Allen Institute, do you plan for a certain number of years ahead, or what does the next 5-10 years look like for your projects at the Allen Institute?
Yeah, that's we already moved on to profiling the human brain, and again supported by the NIH Brain Initiative. So we are moving into a new phase of the Brain Initiative cell census effort. The plan is to finish the Human Brain Atlas in 5 years. So, and then of course it will take a lot of time to understand. I don't think we'll be able to, completely finish data analysis of the data generated from human brain. We'll continue to learn. Maybe it will take another several years to truly understand what's going on in the human brain. But yeah, we have a pretty ambitious plan.
I have another question. As a PhD student, I gradually realized that it is very important to develop insights of what questions are important and what research directions we need to pursue. How do you explore and identify what questions are important and fundamental in neuroscience? Do you have any suggestions to junior researchers on how to develop such insights?
Identifying the right question to address is very, very important. My PhD and postdoc mentors, they were both great scientists, accomplished scientists. I think what they emphasized to me as a mentor or to other trainees in the labs were also very much focusing on asking the right question. For example, they said one of them said that it takes the same amount of time and effort to work on an important question as working on an unimportant question. So it's not like, okay, if you work on something not so important and trivial, it will be easier. It's not like that. You will spend the same amount of effort to do that. And so it's really, really key to identify the right question. How to do that, and that's something that again you learn from your study, from your research. I feel that you really need to have a deep understanding of the field. So do a lot of literature research, read a lot of great papers, and discuss with others. With your mentor, with other students or PIs, go to conferences, just absorbing a lot of the information and knowledge, and then it will help you tremendously, identifying the right question to ask.
I'm also curious, how did you identify the current questions that you are trying to answer at the Allen Institute? Why do you want to understand why cell types are important?
So at the Allen Institute, it's very interesting that we now have a really good model of collective decision-making process. It's not something like, say, I as a director to decide by myself. We convene multiple ideation sessions and workshops and discuss with external experts, and also having discussions with in-house internal scientists about what are the questions that we should address, what are the questions that are important to the neuroscience field that we could address using our unique capabilities that will generate the greatest impact. And we, discuss, present all kinds of ideas, and we did ranking exercises, voting and ranking, always having that at the end of the meeting. So we come up with a ranked order priority list about, what questions we should address. And then after that, we convene multiple workshops, focus on each of those specific areas, inviting experts in those areas, and have further discussion about how to do it. So everybody is aligned behind those goals.
It's a very principled way to bring together, to get the feedback from all the experts and to really decide what the best path forward is. Yeah, I see that as being very valuable and helpful. Because those are major decisions, involve major funding, right? Yeah, we took it very seriously. You've talked about a ton of fantastic, interesting questions and research that you've done and your approach to science. At the 2023 annual meeting, you will be sort of opening up the annual meeting, presenting the Talairach Lecture. Is it a little bit too early, or can you give us a preview of what you think you'll be talking about at that lecture?
Yeah, sure. Yeah, the main focus actually is that our most recent publication that Xinhui mentioned. I'm going to talk about the cell type organization across the entire mouse brain. Yeah, so that paper is going to be the centerpiece of my talk, but I will also have an introduction part talking about our previous work, just give a systematic overview of what we have done over let's say the last 5 years, beginning to address the cell type questions using different approaches, and then focus on the whole mouse brain cell type atlas that we have generated, how we did it, and what we have learned, the principles that of cell type organization that we have learned from the whole mouse brain data. And then, if I still have time, I may zoom in to a particular example, talk about some of the further analysis that we have done for a particular brain region or a particular circuit, just a deeper dive into a particular circuit than just this global grand view of all the cell types across the brain. And that's the last part would be the part that we are still working on and that has not been published or posted on bioRxiv yet, maybe by that time. So yeah, just zooming in, to see how it actually makes a difference, for us to understand the diversity and maybe even how it relates to behavior and function and things like that.
Okay, I have another question regarding DEI. As a major female figure in neuroscience, have you noticed any changes in attitudes towards women neuroscience during your career from junior scientist to senior PI? Do you have any suggestions to create a more open and inclusive research environment?
Yeah, I think over the last 20, 30 years, spanning my entire research career from graduate student all the way to now, I think there have been a lot of changes, positive changes in the research field in DEI, especially in two areas. One is the women representation of women, and then second is the representation of underrepresented minorities, right? So when I started, it was indeed a very much white male dominated field, and the issues that people talk about, I think it was true. I was not the person who was vocal about it. I just tried to fit in as a young student, young scientist, coming from China, foreign country. I just try to fit in, and being able to grow, in the scientific community. But I did feel, and felt very fortunate that my mentors, both of my advisors as well as the faculty members that I encountered, And in both Brandeis and MIT, they were very, very supportive. And also at the Art Institute, of course, I was very supported. I do not have any complaint about, not giving enough opportunity or things like that. But I did have the feeling that, when I went to conferences, giving presentations, or just, the general interactions, the feeling that I had to prove myself, I had to demonstrate that I'm capable and worthy, whereas maybe, one of my peers could be just assumed to be talented and capable, right? People would turn to that person, just respecting and listening to that person more rather than me. And I could be, often ignored, and I had to demonstrate myself to have the ability. I had to work hard in order to catch up, in order to move up. Things are getting better. I think one of the main reasons is that we began to talk about it. And I really admire those, many of the women scientists, really step up to point out this issue and to talk about it and make other people more aware of this. So just by talking about it more, by making these issues more open and, being aware by people then will modify their behavior as well and their attitude. And also on the underrepresented minority front, I think, there were very few, almost none, URM faculty members that I knew about before 10, 20 years ago. And now there are just a lot more. And it's very important. I think, for young generation scientists like you, Xinhui, it's very important for you to see role models, right? And make you believe that this is the place that you belong, right? If there's not me or somebody else who show that you can make it, you can go to that level, you just feel like, okay, this career path is really not for me. I truly believe that role models are very important.
Yeah, I have been looking up to you, so your experience really inspired me to grow up some scientists like you. Yeah, thank you for taking the role model role. Okay, our last question. What do you enjoy doing beyond your research? How do you balance your work in life?
I have to do the management. That's something that I have to do, but my heart is still in science. So I still want to do science, reading papers, writing papers, looking at data and thinking about data. So I really don't have much free time to pursue my personal interests, but I like, things like I like to do. I like nature very much, especially I feel fortunate to be in the Seattle area. It's just the nature surrounding us. It's just so beautiful. So I like, hiking, going out into the mountains, into nature as much as possible. And also, staying on water, try to do kayaking and things like that. I like to, interact with my kids. I have 2 children and they are now, already in college and graduate school. So, but it has been a tremendous pleasure, raising them, just helping them to become, to grow, right, from kids into young adult, now into, their independent life. It was a very, very rewarding journey. And we have very close relationship and just, it's amazing.
Yeah.
Raising kids consumed a lot of my time as well, but it's very rewarding.
Thank you so much for your time, and thank you for your interview. I really learned a lot from you during this whole interview process. We are very looking forward to your keynote at OHBM in July.
Okay, yeah, thank you very much for the opportunity. And it's very, very nice talking to you guys.